And I'm pleased to talk to you today about the field of life sciences and biology writ large, based on my experience leading the Biological Sciences Directorate of the U.S. National Science Foundation from 2014 to 2018. So I start with a cautionary tale from the United States, and it's from the 1930s, and it's in the US described as the Dust Bowl. And the Dust Bowl, you can see some pictures here, was a massive dust storm that ruined the lives of many farmers, many poor farmers who lived in the middle of the United States during this period of time. I'll note that this period of time, the 1930s, also coincides with the US Great Depression. So it was preceded, the Dust Bowl was preceded by science policy-based production of agriculture. The idea of producing agriculture on an industrial scale, but land that was increasingly marginal. The middle of the United States, where the Dust Bowl started, is very arid. It's a part of the landscape that doesn't naturally, is not naturally favorable to agriculture. And you can see some pictures here of agriculture as it took place on this land in that period of time. I want you particularly to note that the most rudimentary techniques of tilling the soil were used, and the soil was very thin. So the land itself is at risk, and when drought happened, that decreased plant cover and resulted in a high plant mortality. And with a high plant mortality, the soil loosened up, and that produced, in turn, these massive dust storms. It was really unprecedented in US history to that time. The soil erosion, you can see the pictures here, was just astounding. It actually occurred at a continental scale, so it started in the middle of the United States. But truly, it was documented, the dust as far as the United States East Coast. So the failure here was understanding how a local event, a regional event, was connected to continental scale environmental impacts. And in my opinion, the entire reach of modern life sciences outside biomedical research has been to connect different hierarchies to understand how local processes can affect the global planet. And certainly thinking in the context of climate change, we're seeing that. So it's not like these things don't happen today. In the top right, you're looking at a satellite image of a dust storm from the Saharan Desert blowing out at continental scale across the Atlantic Ocean. The fact is that climate change is producing the type of events that we saw with the US Dust Bowl with ever increasing frequency. So once again, the cautionary tale here is about thinking at different scales, all right? What happens locally with appropriate feedbacks from the planet can have global effects. And I would say generally, biology is confronting a changing planet, and modern life sciences in particular, outside biomedical research, is looking at the biosphere as it's responding to the forcing function of these massive changes. And we can see that from in the US from the increasing frequency of devastating wildfires in the American West. We're seeing drought that's affecting agriculture. We see modern versions of the dust storms that I was talking about, increasing flooding from the Mississippi River. And the lower right-hand pictures of Superstorm Sandy, which was a combination of a massive hurricane and a winter storm that merged with it, that came out of Canada, that hit the US East Coast in 2012 and was catastrophic in terms of its effects on New York City. So this is what biology is confronting nowadays. The center graph is looking at changes in carbon dioxide in the atmosphere over time. You can see it goes from 1960 up to 2010. The trend has continued. You can see the seasonal bumps that are natural, but the increase in carbon dioxide, a potent greenhouse gas, is producing the very effects that you see in the panel pictures. So the change is ubiquitous. And we see that change, but we don't really understand the details of how and at what level carbon dioxide, increasing carbon dioxide in the air, is producing these emergent effects. I like to think that biology has transformed recently from being somewhat like stamp collecting in the 19th century to being very predictive, to being a quantitative predictive science like physics and chemistry. And this plays out, and this transition in biology is very important because it allows for us to make sense of what's going on with climate change and how that plays out in Earth's biosphere, and make predictions, reasonable predictions of what lies ahead, and therefore make intelligent policy decisions about what to do about what lies ahead. This is a paper about the nature of signatures in the biosphere that relate to critical transitions. That is a way of thinking about the biosphere that is very similar to the way the field of physics and chemistry relate to those more simple systems. The key is making connections across scales. So from left to right, we're going from the level of DNA from the genome to integrated organismal systems to what's going on with entire ecosystems. And that plays out at a continental scale. The goal is to better understand what chemical and physical processes drive this massive planetary change. To do that, biology needs new data streams, for example, from precision phenotyping, linked to proven approaches. And biology needs to be very innovative in terms of new theories that essentially allow us to understand the rules of life, an observation, an experiment, and modeling across scales. So from the level of the genome in a single cell to what's going on with drought across an entire continent like the United States. I think that biology is at a seminal moment. The tools that are available in biology, the ubiquity of those tools, the fact that they are so inexpensive now, is really changing the field. This is a graph of carbon sink for the planet at continental scale, from a paper in Nature Climate Change in 2019. And the point that I'd like to make from this figure is just how vast our ability is to actually look at what's going on with the biosphere at the level of the whole planet. And this is made possible, of course, by remote sensing, but connected to ground truth observations. So, for example, on orbit measurements of proxies for carbon dioxide linked to eddy covariance measurements of carbon fluxes at ground level. That ground truthing allows for maps like these to be produced. And by looking at maps like these, we get a picture of actually the planet's ability to fix carbon from the atmosphere and store it in the form of plant material and animal material, and other fixed carbon deposits across the planet. All right, and we need this to actually understand what's going on with the biosphere going forward. My former directorate just finished the completion of a $500 million observatory, the National Ecological Observatory Network. You can see some schema pictures of it below, but it essentially allows us to build a time series of the North American continent, some 200 data products, very rigorously maintained going forward. And the US will spend $2.2 billion over the next 30 years curating that time series. There's a lot of robotics involved with it, a lot of remote sensing, a lot of humans sampling DNA. But it is a massive effort to actually get a readout at continental scale, extending from Hawaii to Alaska to Puerto Rico, across the American continent, of what's going on with the biosphere. And this instrument is really a first of its kind. You can think of it as a telescope looking at the biosphere of North America. And if we think about the planet as a whole, and we think about the fact that each defined square of longitude and latitude, say kilometer by kilometer, represents some piece of the biosphere, some volume of the biosphere, the ability to actually sample the planet as a whole rigorously, what's going on with it, that's what we really need. And NEON represents a first shot at that across the United States. Another aspect of understanding what's going on with our planet, what's going on with the biosphere, means taking care of the observations that have been made in the past. And we're doing that. So if you think about collections of beetles from the past, right, at the Smithsonian Institution, for example, these biological samples with their DNA in them represent a window onto the biosphere as it was when the samples were collected. And we now are engaging in curation and rigorous data extraction from these samples in a non-destructive way to actually get a clearer picture, not only of what the future might be like or what the present is like with neon, but also what the past was like, to build up a more complicated view. So that's the cautionary tale. Now, let's talk about the real story. Life is ubiquitous on this planet. It is everywhere. It's in the deep ocean, it's in the Arctic, it's under the Antarctic ice, it's even under the crust of the deep ocean, all right? It is everywhere we look for it on the planet, there is life. It may also exist elsewhere in our solar system. We, I think, know from the robotic rovers on Mars, Mars once had water, Mars once had an atmosphere that was more substantial than it has now. Mars may at one time have been a very friendly planet for life. And we may yet discover evidence that life existed on Mars. The same is true with some of the other moons and moons in the solar system, and here are some examples. We are all investing in robots, probes to actually go look close up at what's going on elsewhere in the solar system. So it is to be expected that we'll at least find out what the story is with life beyond Earth, elsewhere in the solar system. And life may be ubiquitous throughout the universe. On the left-hand side, you can see a remarkable radio telescope picture of a developing solar system. This was produced by the Atacama Large Millimeter Array in Chile. It's a collection of radio telescopes that operate together to act as if they were one giant radio telescope. So you're actually looking outside the solar system at this star HL Tau, and you're looking at a picture of a developing planetary system beyond our own solar system. And the tracks you see are actually the developing planets. And we now know that there are many stars that have planets that are at a habitable zone from the star, and the collection of such planets keeps going up and up. So life may be, in fact, ubiquitous throughout the universe. And if you think about life on our planet Earth, it may be the life itself which engaging with the planet produces the conditions for life to exist. And Venus is a planet that is close to our orbit. It is about the same size of Earth. It's completely uninhabitable. The surface temperature is on the neighborhood of 800 degrees C. And a lot of us in the field think that without life on Earth, Earth would be like Venus, with a massive atmosphere of carbon dioxide, runaway carbon dioxide, producing immense amounts of heat. So biology is the story of life, all right? And on the left-hand side, we start with an anoxic world, Earth, just after it was formed. And in the deep ocean, we have these vents that produce heat produce interfaces between redox couples and allow for complex organic molecules to be synthesized de novo. We have life in the sea leading to microbial stromatolites. Photosynthesis develops. Life on the land, life moves to the land, and suddenly we have the great oxygen event where the atmosphere of Earth transitions from being very high CO2, low oxygen to being high in oxygen and allowing for life forms like our own. And high oxygen allows for remarkable biology, like you see here. So one of the major questions for biology is, how did life actually begin? And I think there are two schools of thought here. One is that the metabolic chart containing the Krebs cycle and that is conserved across all of biology contains the most important clue about how life originated on our planet. That idea is that the metabolites themselves, that chemistry, constrained the rest of biology, and that's why that metabolic chart is ubiquitous. It is absolutely conserved across all life forms. And the other is that RNA, in its the RNA polymer and its natural synthesis in wetting and drying cycles in shallow ponds created life on the planet. And I think there's elements of both these stories that may be correct, but the field of astrobiology and how life began on the planet is utterly fascinating and is really important in terms of answering the basic question of whether life may be ubiquitous and self-organizing elsewhere in the universe. Living systems are constrained, right? So as humans, we understand that we have metabolic constraints. We need food, right? There's a mass balance. There's an energy balance. And you can think about this at multiple levels. You can think about it at the level of a cell, single cell, but you can think about it at the level of an organism, or you can think about it at the level of an ecosystem, or the biosphere as a whole. But these constraints are very important. They are both physical and chemical. And really, they are what connects biology to the basic sciences of physics and chemistry. So I mentioned earlier rules of life. And the rules of life is one of the US National Science Foundation's 10 big ideas. And let me talk about rules of life for a little bit. So let's take the game of chess, right? It's a very simple game. It has a defined set of rules. It has a defined chessboard, defined pieces that are constrained in how they can move. And there are, I think, 10 to the 287th power number of possible chess games that could be played, all right? So it's constrained, but even with those constraints, those rules lead to a vast, unbelievably vast complexity, which is the number of possible chess games, right? Okay, that's an example of a rule set. Another example of a rule set is thinking about chemistry and atoms. So we know that the Pauli exclusion principle means that if we have an electron in an orbital with an upspin, the only possible other electron in that orbital has to have the opposite spin, a downspin, all right? And the Pauli exclusion principle, the most simple rule, right, yields the periodic table of the elements, all right? It constrains atomic structure across the universe. And by constraining atomic structure, you get chemistry, you get organic chemistry, you get biochemistry, right? So the most simple rule leads to the periodic chart of the element, leads to biology. That's a rule set. All right. So those are known rules. Let me get to a lesser known rule, a rule that we don't know, but we know must be there. So let's take amino acids, right? So there are 20 there are 20 amino acids that go into proteins, right? And a protein is a polypeptide of something like on the order of 100, can be a lot more, but let's say 100 amino acids, right? So if we just say we've got 20 amino acids that can come together in 100 mers, to form peptides with 100 amino acids, that produces 20 to the 100th power number of possible proteins, right? But there's no such number. I mean, the biological space is way constrained. Even if we take all the known proteins, all right, there's way less than that number of proteins. So something else must be constraining the phase space of available proteins. It's an unknown rule of life, right? And there are many of those. I could go on and on. We can look at every single nerve cell in a zebrafish embryo as the animal is behaving. We can look at the activity of all those nerve cells, but we still don't know the mapping function between the activity of those nerve cells and how the animal is behaving. That's another example of an unknown rule of life. So rules of life, rule sets are really important in biology. And I've delineated some in this slide right here. One very exciting rule set, of course, is the rule set that governs how genotype plays out in phenotype, right? A terribly old question. It's been around forever. But we really don't understand it, other than we know that the genome is important and the environment is very important and it produces phenotype, but we don't understand how that actually happens in detail. And I would say the major challenge for biology, not only basic biology, but especially in the context of biomedical research, certainly in the context of agriculture, is understanding the rule set that goes from phenotype, from genotype to phenotype. I mentioned earlier that technology has radically changed biology. The graph on the right just shows the cost of sequencing and how it's dropped precipitously, and that allows genomics to be ubiquitous. We sample microbiomes, environmental DNA all the time, and it's a very inexpensive process. The dongle in the picture is the nanopore DNA sequencer, plugs into your computer. So the whole technology has become, the technology for molecular biology has gone from being extraordinarily expensive to extraordinarily inexpensive. And it allows us to do biology scale quantitatively. It's allowed technologies like CRISPR-Cas to become a reality. But technologies like that have allowed us to ask new questions, all right? When we are allowed to ask new questions in a scientific manner, all right, that improves our theoretical framework, right? Allows us to build a better scaffolding to understand what's going on with biology, with the biosphere as a whole. And I started off with my cautionary tale about the Dust Bowl and continental scale of the biosphere. You need new technologies to ask questions, to do the science, to actually understand how that plays out. And Certainly, those technologies are not just limited to sequencing. We're looking here at precision phenotyping at an ARPA-E installation in Arizona. All right. Robotically looking at the phenotype of individual crop plants under very specific and modifiable conditions. This is another example of the technology changes that have been made. So, of course, all of that's generating an enormous amount of data. And actually understanding, making sense of massive amounts of data requires two things. First of all, it requires an enormous amount of very sophisticated high performance compute power, all right? And nowadays we use not only supercomputers, but we use machine learning and artificial intelligence on a massive scale to actually make sense of data. In a lot of biology now, we're using that compute and AI to actually do automatic hypothesis production because the complexity of the data is too great to actually produce hypotheses in a sophisticated way, taking into account all the data in the field without the help of machine learning. But we also need ways to make that data available to all, freely available, curated properly with appropriate metadata. All of that's critically important. And this produces challenges. It produces challenges not only here within my country, within the United States, but in terms of international cooperation, it's incredibly important. My own belief is that the problems of climate change, the problems of biology, think the pandemic that we've recently gone through, right, go beyond national borders, and that the data that biology produces now needs to be shared fully, and that means that it needs to be available, curated, its provenance must be understood, and that's a challenge, and it's worth investing in. All of this goes back to the fact that we have this framework theory in biology called the DNA dogma, DNA, RNA, protein, right? And it's a great framework for thinking about biology and making biology quantitative, but it's also important to realize that that is a very simplistic representation of what's actually going on in the biosphere. We look at things like epigenetics, RNA interference. It's an example of how that dogma actually has a much more sophisticated overlay that is on top of the dogma, and that we need to understand also, not only in the context of human disease, but in the context of actually understanding how the system as a whole works. Beyond RNA dogma, we need to go beyond the idea that this notion of the tree of life, that there's been, information passed on through selection, history, and contingency from earlier generations to current generations, and understand that actually you can have horizontal gene transfer between organisms that doesn't depend on this tree at all right? And certainly we see that a lot in forest ecosystems with fungi and trees and the microbiome. And so the original notion of tree of life, rooted in this common ancestor, has gotten considerably more complex, not only with the idea of horizontal gene transfer, but with the discovery that there are other kingdoms, such as the Archaea kingdom, that we weren't even aware of, that were hitherto invisible parts of the biosphere, but are important. So this is all, these discoveries have all been made possible because we can do DNA sequencing, all right? So DNA sequencing on a massive scale has allowed us into windows, into rule sets, rules of life that we were not aware of before, all right? So every field needs a framing narrative. What's the framing narrative for biology? So this is what I think. I think biology is, in fact, the modern engine of innovation. Look at the idea of the mRNA vaccines and how they played out in the pandemic, all right? That was innovation, and it was innovation developed at scale and at speed, and it's made a difference in the lives of lots and lots of human beings. And the example of the mRNA vaccines may be applicable in other fields, in other diseases. But this notion that actually biology can be the engine of innovation, I think, is a very important framing narrative. And thinking about agriculture and thinking about food security for our future planet, it's very clear to me that biology is going to be critical to our ability to feed all of these human beings in a way that doesn't destroy the planet due to eutrophication. You know, the earliest framing narrative for biology, just after Charles Darwin, was the question of what is life, right? And if you think about it, that's not a trivial question, all right? And a lot of very smart folks have thought about that, including the father of quantum mechanics, Schrodinger. But that's where the framing narrative for biology starts, all right? During the 20th century, the arc of the framing narrative moved to the goal of controlling living systems, particularly in the context of human health. I often take my undergraduate students through how the whole discovery of antibiotics changed human life. If you think about the fact that a scratch or a small wound was a death sentence often in the 19th century, and by the mid-20th century, it was a trivial occurrence, all right? So that is an example of how the arc of biology changed over the course of the 20th century. And in our time, here, now getting into the second decade, close to the third decade of the 21st century, I think the arc of biology is now moving into this new science of synthetic biology, all right? And it gets at these rule sets. But certainly, what are the physical rules for cell assembly? What is the minimum genome required to sustain life? How does genome relate to phenome? And how can we use the building blocks of biology to actually come up with really creative environmental applications, such as controlling oil slips or building staple crops that don't require fixed nitrogen fertilizer? So synthetic biology may be a critical, important tool for precision medicine. The whole notion of CRISPR-Cas as a way to edit the genome comes to mind at that. If you think about the mRNA vaccines themselves, they represent a really modern application of synthetic biology. So we essentially go from the alphabet, the genetic alphabet for the spike protein that binds to the receptor for SARS-CoV-2, ACE2. And we take that alphabet and we inject that alphabet into humans to allow our own human synthetic machinery to produce the spike protein that produces immune response, adaptive immune response in us. That's an exquisite example of synthetic biology. I really do think precision medicine is going to be the way of the future. If you think about how we take drugs nowadays to cure diseases, and we assume that one size fits all, at least for a subpopulation, that's not efficient, right? We're all different. And we now have the ability to actually quantitatively look at how we're different. And since we have that information, it makes sense to actually use that information to develop therapies that are tailored, individually tailored for all of us. Another way that life sciences is going to play out in the future. Obviously, we need all hands on deck, all right? So if we exclude women, if we exclude underrepresented minorities, then we are losing out on some of the smartest, brightest folks around, and we need as many smart people as possible. Our world is changing. The story of life continues, and I hope it continues for some time, all right? But that's what we want, that's the goal. I'm going to stop my share. And I want to thank you for listening to my talk. So first, dealing with big data. It is certainly true that biology is all about massive amounts of data. Our genomes are big. That is, the information contained in our genomes is very big. If you look at the number of possible proteomes, that's also a very big number. That is, there are a huge number of proteins that are possible. We talked about protein space earlier. We look at the diversity of organisms across planet Earth, across the biosphere. It's a big number. So whenever we're dealing with life sciences, we're dealing with both complexity and very large amounts of data. And this would be impossible were it not for the advent of computing and high performance computing at a very high performance computing, particularly now with the advent, the addition of artificial intelligence and machine learning. So we inherently, in all of the life sciences now, use the tool of computation as an assistant to handle the fact that life sciences inherently involve massive amounts of data. I would say that the ubiquity of computing, from our smartphones to the laptops that we have in front of us, all of that is what makes possible our ability to deal with large amounts. Now let me turn to the question of genetically modified organisms. So I realize, having in a policy role represented my own country to the European Union in this area on multiple occasions, that there are differences of opinion with regard to this concept. I guess I would stick with the idea that is most prevalent here in the States, that food security is really a huge issue. And as I mentioned in my talk, Earth is 2 billion human beings over its natural carrying capacity. And it's likely to get much larger. So we somehow have to deal with the fact that we have to grow food for all the folks on the planet. And to do that efficiently, to be able to feed everyone without poisoning the planet from, say, excess artificial fertilizers, we have to do something. And the notion of genetically modified organisms, I think, really comes down to the fact that we're simply engineering the process that nature already uses through evolution to modify the genomes of organisms, right? So evolution, and it represents systematic changes in genomes over time, driven by history and contingency, and mechanistically by mutations that are selected for. That represents genetic modification. And so when humans do this in the laboratory to, say, increase the yield of maize or wheat or rice, this is really a process that nature itself undertakes. Now, the engineering driver for nature has to do with reproduction of the organism, right? Whether the engineering driver for thinking about food security has to do with yield, for example, of a crop. But this is obviously an issue for which there are different opinions, and I do my best to be open to all those opinions. But the fact of the matter is, ultimately, when faced with food insecurity, nations do what they got to do. And I think that will go for the planet as a whole. Turning to biomedical research, and in particularly in the context of infectious disease, look, I actually think with regard to COVID, we got off pretty easy. I think the mRNA vaccine technologies were fabulous. They saved innumerable lives across the planet. I sincerely wish China had adopted that technology earlier and successfully, but more successfully than it did. And I think mRNA vaccine technology will have a whole slew of applications for future infectious diseases. If you think about it, mRNA technology, vaccine technology is essentially digital. It means you type a sequence into a computer, and out comes the mRNA that will serve as an information template to train an immune system. So the technology of tweaking and producing mRNA vaccines can be very, very rapid. And their use not only in infectious disease, but in terms of cancers, has a really grand future, in my opinion. Nonetheless, it certainly is true that there are a lot of areas of biomedical research which are entirely frustrating and have seen very slow progress. And I would put my own field of neuroscience, molecular neuroscience, at the center of that. I remember very early on briefing the Obama administration to invest enormous amounts of money in a brain moonshot, and with that notion of fixing Alzheimer's disease and schizophrenia and the like. And I think the progress has been remarkably low, particularly for the input of money that's been put in. And I think probably the problem is, and I don't know if I said this in my talk, but before we can really understand brain diseases, we have to understand how the healthy brain works. And I think the healthy brain is so complicated that represents both a data and a complexity challenge. Finally, I'd like to address climate change. I think climate change and the biosphere are connected, but not in the way that a lot of folks are thinking of. And by that, I mean, we tend to value ourselves and put ourselves at the center of the biosphere. But from a planetary standpoint, that's not necessarily true. What I'm getting at is, I'm sure with climate change, life would evolve in whatever ways to continue a biosphere on the planet. We've had ridiculous amounts of climate change before in Earth's history, far larger than we're seeing now, and life has always survived and adapted. So I think the real problem with climate change is what it's doing to us and our own survival as a species, and that's much more problematic. Gets at this problem of food security, gets at the problem of what happens with sea rise as ice caps melt. I don't really have any easy solutions because I don't see how governments are going to cooperate effectively enough. They certainly don't cooperate effectively enough to prevent wars, for example. So I'm pretty pessimistic about governments cooperating on climate change. I do think it's inevitable that a very wealthy person or a small nation is going to adopt geoengineering approaches themselves, just because it's cheap. It's within the geoengineering Earth's atmosphere is within the range of something a single billionaire could afford. So someone's going to do it, and then we'll see. So I hope that helps. I enjoyed giving you your talk. Take care.